Good morning and welcome to this meeting of the Midwest Comparative Effectiveness Public Advisory Council. We will be starting in 1 minute to allow people time to log on.
It's 1 minute after, so I think we should go ahead and start. Welcome everyone to our Midwest CEPAC meeting today, discussing a really important topic and questions. I'm Reem Mustafa. I'm the chair of the Midwest CEPAC. I'm an associate professor at the University of Kansas Medical Center. I also direct the outcomes and implementation research. And I do not have any financial conflict of interest to declare. At the beginning of every Midwest CEPAC meeting, we like to ensure that speakers introduce themselves and disclose any financial relations with industry. Every member of our Midwest CEPAC panel who is present today has met the ICER conflict of interest policy. And I would like them to introduce themselves and their work and note whether they have any conflicts to declare. I'm going to start by really in the order in random of how they appear on my screen. So let's start by Alan. Alan, would you like to introduce yourself? Okay. Maybe if Alan is not able to, should we go to Nick?
Hi, I'm Nick Bagley, University of Michigan Law School. I have no conflicts to report.
Thank you. Bijan?
Hi, I am Bijan Bora. I am a professor of health services research at the Mayo Clinic College of Medicine and Science. I have no conflict.
Excellent. Angela?
Good morning. I'm Angela Brown, CEO of the St. Louis Regional Health Commission, and I have no conflict.
Thank you. Aaron?
I'm Aaron Carroll. I'm a professor of pediatrics and health services researcher at Indiana University School of Medicine, and I have no conflicts.
Great. Don?
Morning, Reem. Can you hear me?
Yes.
Great, thanks. Don Casey. I'm a physician internist, president of American College of Medical Quality faculty at Rush in Chicago, Thomas Jefferson in Philadelphia, and University of Minnesota Institute for Healthcare Informatics. I have no relevant financial or conflicts or relationships with industry. Thank you.
Thank you. Stacy?
Hi, everybody. Stacy Doucette-Zena, Associate Professor of Health Policy at Vanderbilt University School of Medicine, and I have no conflicts.
Thank you. Ingva?
Hi, I'm Ingve Falketer. I'm a professor here at Case Western Reserve University. I'm a hepatologist, and I have no conflicts of interest.
Thanks. Albert?
Good morning. I'm Albert Wong, professor of medicine at the University of Chicago. I have no conflicts of interest.
Thank you. Jill?
Hi, I'm Jill Johnson. I'm a professor at the University of Arkansas for Medical Sciences College of Pharmacy. I'm a clinical pharmacist, and I provide the clinical support for the evidence-based prescription drug program, a service arm out of our college. I have no conflicts with industry.
Thank you. Bradley?
Hey, good morning. My name is Brad Martin. I'm a professor in the Division of Pharmaceutical Evaluation and Policy at the University of Arkansas for Medical Sciences.
Thank you. Scott?
Good morning. I'm Scott Misek, professor at the University of Health Sciences and Pharmacy in St. Louis, and I have no conflicts of interest.
Thanks. Rachel?
I'm Rachel Sachs. I'm a law professor at Washington University in St. Louis, and I have no conflicts to declare.
Thank you. And I'm going to try one more time. Ellen?
Hi, I'm Alan Balch. I'm the CEO of the National Patient Advocate Foundation, and I have no conflicts.
Great. Thanks to all of the Midwest CEPAC member. We're also very fortunate to have our patients experts and clinical experts with us today. And I would ask you to introduce yourself and again declare if you have any financial relationship with industry or potential influences on judgment when you speak. We will also present this information publicly on slides, I believe. So I will start with Kat Davis Ahmed.
Good morning, everybody. My name is Kat Davis Ahmed. I'm Vice President for Policy and Outreach at the FH Foundation. And I also have familial hypercholesterolemia myself and work with many people who have FH and clinicians. The FH Foundation is a nonprofit 501c3, and we do receive support from multiple pharmaceutical companies, including but not limited to the companies who have products under review today. And of course, we also receive support from individuals and families affected by familial hypercholesterolemia and other inherited cardiovascular risk. Thank you for having me.
Thanks, Kat. Andrea Baer.
Hi, thank you. I am Andrea Baer, the Executive Director of Mended Hearts and Mended Little Hearts. We are also a 501c3, and we receive funding from pharmaceutical and device companies, not exclusive to the two that are being reviewed and looked at today. And we also receive funding from patients and families affected by hypercholesterolemia.
Welcome. Thanks. Keith Ferdinand.
Keith Ferdinand. I'm a clinical cardiologist and the professor of medicine at Tulane University. I have done consultation work, nothing specifically related to bempedoic acid or inclisiran, but I've consulted in other areas for Amgen, Novartis, Eli Lilly, but no conflicts directly related to these two products.
Thank you. Salim Virani.
Good morning, everyone. Salim Virani from Baylor College of Medicine. I'm a professor in the section of cardiology and cardiovascular research. I'm a preventive cardiologist and a health services researcher. I have research support from the Department of Veterans Affairs, World Heart Federation, as well as Tahir and Juma family. I have received honorarium from the American College of Cardiology in my role as the Associate Editor for Innovations for the ACC's educational website, acc.org. Those are my financial disclosures. Thank you.
Thank you and welcome to all of you. We appreciate you taking the time to be with us today. I would like now to introduce Dr. Steve Pearson, President of ICER, who will take us through some introductory slides.
Thank you, Reem, and thanks to everybody. It's still a shame that we can't be together in person for these meetings, where we really appreciate, I think, the importance of being together when we deliberate around issues such as the ones we'll talk about today. But let me give you a brief background to this process today. And the meeting is going to be one that I think that will cover some very important ground. But we want to start with the fact that it's rooted in aspects of what we can do for patients. So why are we here today? Let me just read a quote from a patient with heterozygous FH. I have not had any cardiovascular events so far, but I do worry every day that I didn't do enough, early enough in life to prevent heart disease. I almost lost my father at age 57 when he had a sudden cardiac death in the middle of a tennis tournament, but we always expected that to happen, not if, but when. And when you hear that kind of quote, you recognize that often why we are here today is because there are always going to be really important needs that patients' families have regarding their health and the way that their health affects their lives. And one of the reasons that we're here today is because when a new treatment, like the two that we'll be talking about today, are approved by the FDA, there's really a sense of celebration because there is that unmet need. There's a sense that we have new opportunities, new options to help patients, particularly those who haven't been well helped by other available treatments. And it also represents the culmination of a lot of hard work by scientists, by clinical researchers, by patients and families who participate and increasingly help guide the clinical trials, and by the company that has taken the time and the risk to bring these products forward. So with that sense of celebration, though, there's also the sense that, from day one, there are going to be questions about coverage, questions about who's going to pay what, how much out of pocket for patients, how much by insurers and employers. And we do know that from day one, often patients can have difficulty accessing treatments when they come into the healthcare system. That addresses issues that they have around coverage eligibility, and it has to do, as I mentioned, with costs. And so as we kind of take stock of this sense again of celebration and achievement and opportunity, there's a sense that we need to put it into context. And another reason is not just because of cost, but because of what happens, if you will, by extension to other patients and others in our healthcare system more broadly. And one way to bring that home is just, again, to put a face on it, if you will. And we could choose any number of faces, but this is one that was in the public domain. And the issue arises, what happens as we welcome new treatments, but are always worried about adding new costs as well? Well, in some ways, it leads to the tension that for many people and families, there may not be enough money for them to afford their health insurance. And this was a story about the Whitman family in Alaska, who are not poor, if you will, by formal definition, but now tend to fall into that gap that we have between the subsidies in the Affordable Care Act and the ability of patients to afford the health insurance plans offered to them. So this family found that their premiums were getting close to $2,000 a month for a $10,000 deductible plan. They really didn't feel like they could afford that given their other housing needs and other family kind of requirements. And so they went without health insurance. They felt they couldn't afford it. And during a couple of years, the story relates how the kids actually got hurt a couple times and the family really struggled with whether they could go to the emergency room and whether they were creating long-lasting problems by not treating one broken bone, one concussion. So this tension is not just around money, it's around the fact that people, real people suffer and actually have health harms that can arise if we don't do the right job of making sure that we can create a sustainable and affordable health system for everybody, not just those at the top of the scale, but for everybody. So in a pictorial form, you could say that we really do want to focus today in many ways on the patients and the families in front of us, who have ASCVD, who have heterozygous FH, who need and will welcome these new treatments. But we have to remember that around them, and often not as visible as we would want, are other families. And apologies that these icons don't show other families in all of their diversity, but the colors are meant to imply that we are a diverse country, and there's a diverse sense of who we are and how we want to be able to keep our focus on the patients in front of us while making sure that we do so in a responsible fashion that allows us to care well for everybody. So how do we do that in terms of this report and in terms of this meeting? Well, you've met the members of the Midwest CEPAC. I need to tell you a little bit now about ICER, which serves in many ways as the secretariat for this process and is the convening host. First, our financial funding and therefore conflicts of interest. This is an updated picture that we keep on our website. And as you'll see that our overall funding comes predominantly from nonprofit foundations, the chief of which is the Laura and John Arnold Foundation. We also receive funding from the California Healthcare Foundation. And recently we've also received funding from Commonwealth Foundation and several others. For a separate kind of process and a kind of set of activities that we run, we take direct contributions from health plans and provider groups, which you see represents about 9% of our overall revenue, and from life science companies at around 12% to run our ICER Policy Summit and other non-report activities, the activities that do not come into play in the research and these meetings that we hold. We also have a variety of different government contracts at around 10%, and a very small number of other sources. So how was the ICER report developed? So it starts more than 8 months ago with a phase that we call scoping. And that's where we turn to patient groups, to clinical experts, to the manufacturers and other stakeholders, and really try to get our bearings around what the key questions will be around the use, the role in therapy of these treatments, and around what the important questions are about the evidence that we should hold in mind. We then go and do a evidence analysis of the available evidence, seeking new evidence if we can get it from sources that might not be published yet, from the manufacturer, from patient groups and others. We also launch a cost effectiveness model, which uses that evidence to help look downstream. And we often work with external academic collaborators, as we have done in this case. The report goes through several different cycles of public comments and revision. And in this case, we were also fortunate to have 3 expert reviewers whom you've already met, Kat Davis-Ahmed, Dr. Ferdinand, and Dr. Virani, who all have had a chance to look at the draft report and give us important feedback. So speaking of that report, I'd like to mention how it's structured to support the voting and the public discussion that we'll have today. And one way that we do that is to show our value assessment framework kind of graphic, which really starts with the goal that we think that everybody at the table here and in the future has, which is we all want a healthcare system that is featured by the combination of having fair prices that really does drive fair access and supports future innovation. And we think that decision makers really need to think about 2 major branches, if you will, of information to help get us to that future health system. One we term long-term value for money, and the other is short-term affordability. And we spend really most of our time thinking about the long-term value for money, but we will talk later in the policy roundtable, certainly around short-term affordability, because that does arise sometimes, even when we have high-value treatments over the long haul, we might have difficulties affording them in our health system in the short term. We want to talk about that tension and how to manage it. But let's talk first about long-term value for money, because that is a complex, if you will, idea. And it's structured in our report to have kind of different components as we think through them. And here again is a picture of how those can look. At the base of it, we really want obviously to think about value for money, starting with, can treatments help us live a longer life? Linked to that, hopefully, is the idea that whether we are or aren't able to live longer, can a treatment help us by returning our function, by helping us be healthier, by producing fewer side effects than other treatment options, all the things, again, that might not lead to a longer life, but that can improve our lives. We want to make sure that when we're looking at the cost, if you will, of something new, we also want to talk about total cost throughout the healthcare system and even beyond, and how does that cost offset any new treatment? How does its cost offset future costs? Like, could it prevent hospital costs or doctor visit costs in some way by improving our health? Then in a different color, because sometimes they're harder to measure quantitatively, but no less important, we want to talk about benefits beyond health, and you'll hear about those in the votes that will be taken. And we talk about special, either social, or some people would frame them as ethical priorities that we have around different conditions, given their background, their historical background, and the certain aspects of the condition that we might want to favor in certain ways. So our report is structured to have information in all these areas. We will talk through these during the meeting today. And just to get very specific about how this relates to the price that often gets kind of highlighted in our reports and in the discussions, how do we integrate those different components of long-term value for money when we're talking about price? Well, we usually start with the idea of cost effectiveness, which comes from the model. And again, that links what was at the bottom of those kind of 5 components. And we can come up with the price that a treatment would need to be priced at in order to reach a certain threshold of cost effectiveness. And recent research in the United States continues to solidify the idea that if we want to find that right balance, where at least where we are introducing new treatments and pricing them in a way that we are not ultimately creating more harm to families like the Whitmans and others, that we should really seek to be right around $100,000 for a new quality adjusted life year gained. So you could say, well, if we stick to that as our ceiling price at which we don't want to go beyond, then, okay, we're going to start there, but we're going to have a range between maybe 50,000 for quality or equal value of a life year gained, which we use. And that's the range. And then we would use our consideration around these other benefits and special priorities to kind of help us kind of manage someplace within that range where we might feel that the pricing was ultimately ideal. We could do it that way. ICER, for historical reasons, partly because the United States is continuing to kind of get itself up to speed with cost effectiveness and to understand how these other issues can be factored in, we currently still use a range that's higher than that. So we actually start with our value-based price benchmark at 100,000. We extend it up to 150,000 as a threshold, but we still, I think it's important to still think about the fact that these benefits beyond health and special priorities should serve an important function in our thinking around what we really mean by value and how we would price to it. So that's kind of the background, kind of conceptual and to a certain sense structural to the report and to the discussion for today. Here's the agenda where we'll start with presentation of the clinical evidence, then the cost effectiveness information, followed by public comment and discussion. During all that time, we'll be fortunate to have our patient experts and clinical experts available for the CEPAC. Then we'll have a relatively brief lunch break to come back to complete the deliberation and have the votes on clinical effectiveness and value. And then we will kind of cap, if you will, the day with a policy roundtable at which we'll talk more about how we actually translate this information about effectiveness and value into the real world. What does it mean we should do around pricing? What does it mean we should do, or what are the options around coverage criteria, and how to choose which patients and how to engage them in shared decision-making around the best options for them? And what kinds of future research do we need to come back in a couple of years and do the best we can for patients and the health system? We'll culminate with some final reflections from the CEPAC, and we'll be done by 4 o'clock Central Time. So that's the layout for today. And so with that, let me hand it right off to Dr. Grace Lynn, who will lead you all through the discussion of the clinical effectiveness information. Dr. Lynn?
Thank you, Steve. Good morning, everyone. My name is Grace Lynn. I am an associate professor of medicine and health policy at the University of California, San Francisco. And I am contracted with ICER to be the evidence author for this review. Next slide, please. I'm joined in the clinical effectiveness section by my colleagues Jane G, who is also an associate professor at UCSF, Felusho Agbula, who's the vice president of research at ICER, and Avery McKenna, who's a research assistant in the evidence synthesis division of ICER. Jane and I both receive funding support from ICER, and collectively we have no other conflicts of interest relevant to this report. Next slide, please. So as Steve mentioned, we're here today to discuss atherosclerotic cardiovascular disease, which is, as you all know, a common and deadly disease. The term ASCVD is a general term, but includes things like coronary artery disease, stroke, and peripheral vascular disease. As you may know, this is the most common cause of death in the United States. And high cholesterol is a major risk factor for development of cardiovascular events. Now, high cholesterol can be something that is familial. And in this report, we'll talk about familial hypercholesterolemia, or FH, which is the most common associated genetic disease with high cholesterol. And it results in premature ASCVD and a high risk of cardiovascular events. Now, there are two types of FH, homozygous FH and heterozygous FH. In this report, we'll be focused on heterozygous FH, which is the most common form of FH. And when I say FH or HeFH, this will refer to the heterozygous FH population. We also know that there are disparities in terms of ASCVD. Black men and women are disproportionately affected compared with their white counterparts, for example, and this will come up throughout, I think, our discussion today. Next slide, please. So how do we manage high cholesterol for our FH and secondary prevention ASCVD populations? Well, if we go back to clinical practice guidelines, they recommend treatment with a high intensity statin to lower LDL cholesterol or low density cholesterol, which is the bad cholesterol, by at least 50%. So we have both a percentage reduction, and then we also talk about numbers. So for this population, which is a very high risk population, if the low density cholesterol remains above 70 milligrams per deciliter, which is set at sort of the goal, it's reasonable to add ezetimibe to the high intensity statin, which you start with, and then a PCSK9 inhibitor. For the higher risk population, for example, the FH patients, for primary prevention, the bar at which you start adding medications to lower LDL is a bit higher at 100 milligrams per deciliter. But again, the next steps would be ezetimibe and then PCSK9 inhibitors. Just of note, what I've spoken about are the American guidelines from the American College of Cardiology and the American Heart Association. European guidelines actually recommend a lower LDL target of 55 milligrams per deciliter. So that's just, if we put it in a world context, there are different LDL targets. Now, the other thing that you'll hear during this presentation is what we call statin-associated side effects or statin intolerance. These are adverse events, for example, muscle aches or lab abnormalities that are related to statin therapy that lead to a lower dosage or discontinuation of the statin. In the clinical trials, this is often defined as an inability to tolerate at least 2 different statins at moderate doses. And its prevalence varies. Estimates vary from 5 to 20% of patients have what we call statin-associated side effects or statin intolerance. Now, because the clinical trials and the literature still use the term statin intolerance, that is the term that we will use today to describe this phenomenon. Next slide, please. So as Steve mentioned, one of the things that we do while we put together this report is talk with patients and patient groups. This slide summarizes what we've learned from patients as we've been discussing this topic with them. And they fall roughly into 3 buckets. The first one is awareness. Under this bucket, we've learned that FH in particular is underdiagnosed and undertreated. And because these patients have a longer lifetime exposure to high cholesterol, this underdiagnosis and undertreatment leads to a greater impact on their life. We've also learned that women often have missed and delayed diagnoses as well. Another bucket is access and affordability. What we've learned is that particularly for the newer therapies, prior authorization and step therapy can make access difficult for patients and lead to potentially high out-of-pocket costs, particularly patients without insurance or with high deductible health plans. And lastly, there was a lot of discussion about health equity. As I mentioned in my prior slide, racial and ethnic minorities bear a disproportionate burden of ASCVD. And there are also disparities in treatment where women and minorities don't reach their LDL goals as often as their male white counterparts. And also we heard that clinical trials are not diverse in terms of gender or race and ethnicity. So these things that we've heard from patients underpin a lot of what goes into our report. Next slide, please. So today we're going to discuss 2 new drugs for the treatment of high cholesterol, bempedoic acid with or without ezetimibe, or inclisiran. And we're going to look at the clinical and cost effectiveness of adding these drugs to maximally tolerated oral lipid lowering therapy for lowering cholesterol. We're going to focus on specific patient populations, as I mentioned, the heterozygous FH population and patients with established ASCVD or patients who've already had cardiovascular events. We have some subpopulations, and that is the HeFH patients with and without ASCVD. Patients with established ASCVD at higher risk, for example, patients who've recently had a myocardial infarction or a heart attack, and then patients with statin intolerance. And as I go through the data, you'll see we've tried to focus on these three groups. The comparator in this analysis are maximally tolerated oral lipid lowering therapies, or what turns out to be the placebo arms in the trial. So as we go through, that is what, when we talk about, for example, LDL lowering, that's the comparator. Next slide, please. So we're going to start with bempedoic acid. And bempedoic acid comes in two forms, either by itself or in a combination pill with ezetimibe. And this is a drug that's an ATP citrate lyase inhibitor. It's a mouthful to say. And that's an enzyme that works in the cholesterol synthesis pathway. So what bempedoic acid does is block this enzyme, then reduces cholesterol synthesis through this mechanism. Now, it's important to note that this acts upstream of the statin pathway, and that will become important when we talk about patients with statin intolerance. This is a once daily oral therapy, and it's approved, was approved by the FDA in February of 2020. Next, please. Next slide, please. So there were some key clinical trials that we looked at, and there are a group of trials called the CLEAR trials, and then a 4-arm study of bempedoic acid ezetimibe combination pill that I'll talk about. So there were 2 studies, Clear Wisdom and Clear Harmony, which looked at bempedoic acid alone versus placebo. And this was in the established ASCVD population with some minority of FH patients. Clear Serenity and Clear Tranquility were somewhat smaller randomized trials that looked at bempedoic acid versus placebo in the statin intolerant population. And Ballantine 2020 was a randomized trial that was 4 arms that looked at bempedoic acid ezetimibe combination. There was a bempedoic acid alone arm, an ezetimibe arm, and a placebo arm. And they also enrolled patients with ASCVD and or FH. They also include some statin intolerant patients, and these patients actually had a relatively higher baseline LDL than some of the other trials. Next slide, please. So here are the results of the bempedoic acid trials, and then the results from a meta-analysis that our team did. So next, please. You can see that for bempedoic acid alone, and that would be these 3 trials at the top. And I will note here that Ballantine 2020 appears twice. Because that was a 4-arm trial, we used separate arms to come up with estimates. So in this case, we had a bempedoic acid versus placebo arms of that trial to come up with our estimate. And you can see that at, the primary outcome was LDL reduction from baseline to week 12. And in this, in these 3 trials, that was around 18% roughly. Next, please. The next 2 trials, the Clear Serenity and Clear Tranquility, these are the statin intolerance trials. And you can see that there's an increased amount of LDL reduction, somewhere in the 24% range. And there's actually a plausible mechanism for this increased reduction in the statin intolerance patients. Remember a couple of slides back, I explained that bempedoic acid works in the statin pathway. It works above the statin step. And so the theory is that if you're not on a statin and you're not already blocking some cholesterol synthesis, that you in fact will have the ability to block more cholesterol synthesis. And thus people who are not on statins or who are on very low dose statins may have a larger decrease in their LDL. Next slide, please. So for our subpopulations, we see that for the FH population, again, this was a minority of patients in the trials, 1 to 5%. There was a possible greater amount of LDL lowering in the FH population, but because of the small numbers, this difference wasn't significant. And so I haven't presented the numbers here, but it was somewhere in the neighborhood of 20 to 22%. Our second subgroup was high-risk patients with established ASCVD. These patients were excluded from the trial, so we actually did not find any data for this population. And our third subgroup was the statin intolerance population. And they, again, showed a greater decrease in LDL-C than the overall population. And you can see there 24 compared to 17%, and this was highly statistically significant. Next slide, please. In terms of clinical outcomes, so there were very few clinical outcomes recorded across the trials, fewer than 200, but these trials weren't set up really to look at clinical outcomes. There are ongoing outcome studies that are whose results will be presented next year, I believe. Next. You can see here that though, even with a small amount of events, that non-fatal MI and major adverse cardiac events, which is an endpoint including cardiovascular death, non-fatal MI, non-fatal stroke, coronary revascularization, and hospitalization for unstable angina, that these look like that the bempedoic acid would reduce these events, though not statistically significant. Next, please. But you also see in this slide that although there are small numbers of events and the confidence intervals are wide, that there may be some signal that there was increase in mortality. Now, I will say that when you look at the mortality data, that the mortality includes cardiovascular events, but also non-cardiovascular events such as cancer deaths, and all the adjudicated mortality events were thought not to be related to the study drug. Next slide, please. In terms of other adverse events, there were more adverse events and discontinuation due to adverse events in the bempedoic acid arms of the trial versus placebo, but most were mild to moderate. The two that I'll bring your attention to are uric acid and gout. There was a 4-fold increase in uric acid in the trials and a 3-fold incidence of gout, higher in patients who had a history of gout. The other adverse event that I will bring your attention to is tendon rupture. There were 11 patients, or 0.5% of patients in the bempedoic arm who experienced tendon rupture compared with none in placebo, the placebo group. And we thought that and this is something that was mentioned in the FDA labeling as a significant side effect. Next slide, please. Now, in terms of some uncertainties in the data and some controversies in the data for bempedoic acid, our data are limited to short-term LDL-C lowering. As I mentioned previously, the primary outcome was 12 weeks, LDL lowering at 12 weeks, and these were in selected populations, and there is not yet outcome data. The drug may offer greater LDL reduction in statin intolerance patients, although again, because we don't have outcomes data, we don't know if this translates into a greater reduction in cardiovascular events compared with the general population. And then it's unclear how significant the risk of gout and tendon rupture will be in the real world, but these were significant adverse events that were seen in the clinical trials. Next, please. So what this brings us to is the evidence rating for bempedoic acid. And we felt like, based on the fact that we had a moderate degree of LDL lowering in the short term, and that we had limited data in the FH population, and there was a risk of moderate to severe adverse events, we gave bempedoic acid a C+ rating, which equates to a moderate certainty of comparable or small health net benefit overall. Next slide, please. So that wraps up our discussion of bempedoic acid, the evidence for bempedoic acid. I'll move on now to inclisiran. Next, please. So inclisiran is a small interfering RNA agent that inhibits hepatic PCSK9 synthesis. And what you can see here is that leads to less LDL receptor degradation, so then clears more LDL. Next, please. that differs a bit from PCSK9 inhibitors, which you may be familiar with, in that PCSK9 inhibitors work outside the liver and block PCSK9 rather than inhibiting its production. Inclisiran is delivered as a twice yearly subcutaneous injection planned for healthcare settings. And as Steve mentioned, the FDA approval for inclisiran has been delayed, not because there are any questions about the clinical evidence for inclisiran, but because of COVID, the FDA has not been able to inspect manufacturing facilities overseas. The evidence that I present today on inclisiran will be the evidence that the FDA reviews during their process. Next slide, please. So the key clinical trials for inclisiran, there were 3 of them that we reviewed. Orion 9, which was exclusively in FH patients on maximally tolerated statin and ezetimibe, and about a quarter of those patients had ASCVD as well. Orion 10 and 11, which were patients with ASCVD either established or with ASCVD risk equivalents like diabetes with an LDL greater than 70. They had roughly 5 to 10% patients had statin intolerance. And you can see they had baseline LDL-C of about 100. Next slide, please. So the trial results are actually fairly consistent across the 3 trials. The primary outcome was baseline reduction in LDL from baseline to day 510. And the trials found an approximately 50% reduction in LDL across the board. And you can see each trial in the different subpopulations there. But taken together, we came up with a summary estimate of a 50.5% reduction in LDL-C for the primary outcome. Next slide, please. In the subpopulations for FH, Orion 9 was an FH trial, and again showed a similar LDL lowering to the overall population. For a high-risk established ASCVD group, again, these patients were excluded from the trial, so we do not have any data specific to this population. And the Orion trials gave us a small amount of data for statin intolerant patients. Less than 10% of patients in the trials were considered statin intolerant, and they again had a similar LDL lowering to the overall population. Next slide, please. In terms of clinical outcomes, next. Here we see that although, again, these trials were not powered to look at clinical outcomes, and clinical outcome trial, a 5-year clinical outcome trial is ongoing and scheduled to be completed in 2024, you can see that even with the few numbers of events, that inclisiran had a significant decrease in a composite outcome of cardiovascular events, including mortality, cardiac arrest, non-fatal MI, or stroke. Next, please. You can also see that in terms of mortality, there was basically no change in mortality between the inclisiran and placebo groups. Next slide, please. In terms of harms for inclisiran, in the clinical trials, there are very few serious adverse events. The adverse events led to a slightly higher discontinuation rate in the inclisiran group. And the most common adverse event was injection site reaction. 5% of the inclisiran group had an injection site reaction versus very few in the 0.8% in the placebo group. Next slide, please. So in terms of inclisiran, to summarize, inclisiran led to a substantial LDL lowering similar to, in scope to the PCSK9 inhibitors. But again, there aren't any outcomes data. And we don't know if the reduction in cardiovascular events will be closer to those of statins or to those of PCSK9 inhibitors. So that's an outstanding question. And then the trial populations were limited. Again, we didn't see very many statin intolerant patients in the trials, and then there was a lack of racial ethnic diversity in these trials as well. Next slide, please. So this leads us to the ICER rating for inclisiran, and because of the substantial lowering of LDL and very few safety concerns, and because of it's similar in mechanism to PCSK9 inhibitors, which have a demonstrated long-term efficacy and safety record, we gave inclisiran a B+, or that equates to a moderate certainty of at least a small net health benefit in this population. Next slide, please. Some potential other benefits and contextual considerations. Fewer cardiovascular events that lowering LDL may lead to may have a greater impact on the FH population because, as I mentioned earlier, they have earlier events and have a longer time horizon for events. Fewer cardiovascular events may also reduce caregiving needs. In terms of mechanisms of delivery, the combination of bempedoic and ezetimibe may decrease pill burden and increase the use of ezetimibe, as that drug is in the real world hasn't been used as much as maybe should be. Inclisiran has an extended dosing interval compared with PCSK9 inhibitors twice a year rather than once or twice a month, and that may impact adherence. And the availability of more effective therapies may impact health equity. As I mentioned earlier, women and minorities are less likely to be treated or reach LDL goals, and the availability of more therapies may impact that. Next slide, please. Once we published our draft report, we received some public comments, and this slide summarizes those. I talked a lot about health inequities as a major concern already, and that in terms of access to care and treatment, disparities in clinical trial representation, and the fact that patient, clinician, and structural factors in our healthcare system and our society contribute to these disparities. We've talked a bit about FH and it being underdiagnosed and undertreated. And then, as I mentioned earlier, the use of ezetimibe in the real world is low. And the ability to combine that with another effective treatment may increase utilization of ezetimibe. So that concludes the clinical effectiveness section of this presentation, and I'm glad to take any questions at this time.
Well, Grace, thank you very much for this excellent presentation, really highlighting the main points in a complex report. Thank you. So I would like to just ask the Midwest CEPAC members if anyone has any question, and I see that Don already has one. Don, please go ahead.
Sure, Dr. Lin, thanks for a great presentation. Just a couple of thoughts here relative to statin and statin intolerance. One is that, statins have been demonstrated to have additional effects besides lowering LDL, right? Anti-inflammatory effects and biologic impact on atheromata. And so the first question is whether you're aware of those effects with the two agents. And the second, well, I'll wait for the first one and then I'll just have a quick follow-up.
Sure. That's a great question. Those things, while there were some impacts of the drugs on, for example, a high sensitivity CRP, the inflammatory markers, they didn't look specifically at the outcomes that you are referring to, those extra statin effects. So while we could theorize that, for example, high sensitivity CRP might be related to, that the inflammatory process and these drugs might reduce that, but we can't make any firm conclusions about that.
Thanks. My other quick question is myself, fortunately not having heterozygous FH, but having an elevated LDL, have been challenged with statin intolerance for about 15 years. And I guess the question is, given that I finally resolved it with some persistence, whether there's any expectation of continuing to try statins after the use of these things. I know it takes a lot of work for some patients, myself included, but I'm just wondering if that was considered a perfect substitute or whether it's, in other words, it doesn't seem to me to be an either or. And I'm wondering if we're losing that message.
Thank you for that comment. Yeah, the statin intolerance population is a difficult one because it really is very patient and clinician dependent. And perhaps actually this would be a good chance to turn to one of our clinical experts, Dr. Varani or Dr. Ferdinand, to comment on how they view the use of these drugs in the context of statin intolerance.
Or, I mean, I could start. So good morning, everyone again. I guess when we look at statin intolerance, it's a very varied population, of course. And exactly as you pointed out, Dr. Casey, it is not a one size fit all, and there is some temporality to it as well. Generally speaking, if we look at large registry data, almost 50 to 60, and in some cases up to 70% of the patients who initially are labeled as having statin intolerance, or the guideline recommended term statin associated side effects, they are able to tolerate some form of statin therapy. Of course, there are always patients, and then there are quite a lot of patients who are not able to tolerate any form of statin therapy despite trying multiple statins. But generally, that is the ballpark based on large registry data. Of course, this is an area where we don't have any randomized control trial data.
Yeah, to the best of my understanding, the term statin intolerance is really not the best appropriate term of art. And that is because when you really look inside of randomized trials, even with persons who have defined themselves as statin intolerance, and then they're randomized to a statin or placebo and crossed over, many times those persons who said they were statin intolerant are able to take it in a placebo-controlled trial. That being said, I agree with Dr. Varani that it's statin-associated side effects or statin-associated effects. That might be a better term. It doesn't roll off the tongue as easily, but it describes what really may be happening. And in many cases, the patients will have muscle aches, arthritis, osteoarthritis, and say it's a statin. The physician then tries them on another statin. They say, no, I can't take it. And they're defined as statin intolerant. I'm not really sure that that's a clear way of looking at that. We have seen patients who we deem to be statin intolerant who can take a statin. But on the other hand, in the clinical practice, and I still see patients, it may be as many as 10 to 15% of patients will absolutely refuse to take a statin. Whether or not it is definitively statin intolerance, I think from a functional point of view, an operational point of view, about 10 to 15% of patients can't take statins.
Oh, well, thank you.
Reem, I just wanted to say, though, I appreciate that it still would be potentially a good idea, right, to, if someone was so-called statin intolerant, to perhaps continue trying to see if you could overcome that in the face of using these alternative therapies?
Yeah, we try. One of the problems is that it's LDL that's the surrogate for lowering the risk. And many times you say the least tolerated dose of a statin and alternative dosing, which is off-label for some statins. But if you're not getting the LDL down in a high-risk patient, 40, 50%, then you probably are not going to get to any sort of goal of less than 70 of the Europeans for very high risk, less than 55. So it's a conundrum. Statins are very, very beneficial. Some people can't take them. Then you say, well, we're going to try every other day, or we're going to try this statin, a milder statin, one that's water soluble. And you say, okay, good, we're on a statin. But now you've moved completely away from getting the patient on the evidence-based outcomes of a high-intensity statin or reducing the LDL 50%. You've thrown that out the window just to say, I got you on a statin.
Yeah, well, thank you so much. This is such an important point and an important group that I'm sure we'll have more discussions about. I'm conscious of time. I'm going to allow 3 more questions, and then we will have time later for further questions. So I'm going to ask Brad, you have your hands raised.
Yep. All right. Hey, thank you. And this is really a follow-up from the discussion we had last week about the relationship to the surrogate and the outcomes. And this is to help me evaluate the model. The model applied a constant rate ratio to the change in LDL levels. Is that consistent? Like, and I guess where I'm going at is it the same if taking somebody from 140 to 130 LDL level, the same as somebody going from, 80 to 60 in that. And I just wanted, I know it's more of a modeling question or interpreting my model, but when you hear the clinical experts on that point.
Good question. Dr. Barney, do you want to go ahead?
Well, I can take that. That's actually a very important question. So it may take me a minute to explain, but I think it's an important one to consider. So what we know based on large body of evidence is that for every millimole reduction in LDL cholesterol, about 39 to 40 milligrams per deciliter, we have about 21% reduction in major vascular events. Now, remember, that's a 21% relative reduction in major vascular events. For a patient who starts out with LDL cholesterol of 160, 170, of course, higher than the US average, if a person is one standard deviation out, their baseline event rates are also higher. So for the same amount of relative risk reduction per 40 milligrams per deciliter, the absolute reduction will be much higher for a patient who has a higher event rate. In general, patients who are in that 150, 160 range have a higher baseline event rate compared to a patient whose baseline LDL cholesterol is 100 to 110. We know the mean levels of LDL cholesterol in the US population are somewhere around 112 or so if you look at the most recent AHA stats update that just came out last week. So for a patient who's in that 1 or 2 standard deviations out of that, they get the same relative risk reduction with 1 millimole reduction in LDL cholesterol, but the absolute reduction you get is higher. It's the same thing that when you give statin therapy to a patient with diabetes, they get the same amount of LDL cholesterol reduction. They get the same relative risk reduction with statin therapy, but guidelines want us to treat those patients more aggressively because in absolute term, that becomes a larger effect size.
All right, thank you. That's helpful.
Just again, for the sake of time, I'm going to actually ask Kat to comment, and then we'll come back to more questions from the Midwest CEPAC members during deliberation. Kat.
Thank you so much. I look forward to talking about the statin intolerance question a little more later because I think it is so important. But I wonder for Dr. Virani and Dr. Ferdinand, I heard Dr. Lin say that there's a difference in the MACE reduction between statin treatment and PCSK9 treatment, which would be potentially comparable to the inclisiran. I wanted to understand if that is the case, if the impact on the MACE is different with the LDL reduction, depending on whether it's reduced by statins or reduced by PCSK9. If I understood what you said, Dr. Lin, thank you.
Yes, that was you're correct, and I did mention that. And so Dr. Ronnie or Dr. Ferdinand or Dr. Kazi, if you wanted to go into that a little bit.
Yeah, I mean, I don't know if anybody else wants to take it. I think the issue we have is that if you look at the PCSK9 inhibitor trials, Kat, I mean, it's a pertinent question. I don't think I have a clear answer. I can give you some ideas and some of the things that are floating out there as to why we would have it. We of course saw a 15%, about 15% relative risk reduction with the PCSK9 inhibitor trials, whereas we would expect a much larger relative and much larger absolute risk reduction. The issue we have with the PCSK9 inhibitor trials, whether you look at the 2 outcomes trials, we have Fourier or Odyssey, our median follow-up was 2.2, 2.8 years, right, for those 2 trials. Whereas for statin therapy, our average follow-up, even in secondary prevention, is much higher, more in the order of 4 to 5 years. And we do know that you do accrue benefit over time. So that could be one of the reasons that could explain that why we didn't see as much of a reduction in these trials compared to some of the statin trials. So we're hoping that with both bempedoic acid and inclisiran, we would have a longer follow-up in the patient, so we actually realize the full benefit that we expect to see with just LDL cholesterol lowering.
Thank you very much. Just again, we'll come back to more questions, but let's go to Kazi, please, and to present about the modeling.
I think Dr. Ferdinand started to say something, but was on mute. If you want to finish that thought.
Yeah, the other thing was that the statin trials were older trials, so they had less intensification of control of risk factors, whereas the PCSK9s were built on the platform of optimal statin therapy.
Great. Good morning, everyone. I'm Dhruv Kazi. I'm an associate professor of medicine at Harvard Medical School, the associate director of the Smith Center for Outcomes Research in Cardiology, and the director of cardiac critical care here at Beth Israel Deaconess Medical Center. And so my perspective on these drugs is twofold. One is that I'm a health economist. I'm interested both in outcomes and costs, but also as a clinician, I'm seeing the most devastating consequences of atherosclerotic cardiovascular disease when they show up in the CCU. Next, please. I have, this work was funded by ICER, but I have no other conflicts to disclose. Next. So we set out in this modeling exercise to evaluate the cost effectiveness of two drugs, bempedoic acid and ezetimibe in combination, or inclisiran, each separately compared with maximally tolerated statin therapy plus ezetimibe for the secondary prevention of ASCVD. And in this secondary prevention population, we're looking at the general population of individuals with pre-existing ASCVD, or those who have pre-existing ASCVD and also carry a diagnosis of heterozygous FH. So in contrast with the clinical discussion that Dr. Lin just led, where she was referring to the universe of individuals with heterozygous FH, when I say FH, I mean individuals who have both FH and pre-existing ASCVD. Next slide. I think it would be helpful before we dig into the model to talk about three caveats. The first is that for each of the new drugs, we're comparing them with usual care and not to each other. As you've heard already, there are no outcomes trials available yet for either drug. So we're making the assumption that LDL reduction with the use of these therapies will result in lowering of cardiovascular events, which is what we truly care about. And we got quite a bit of feedback on this. We are not making a case that step therapy with ezetimibe is right for every patient or in every circumstance. What we're saying is that given the guidelines, the value of the drugs should be determined by the value they bring to the patient in addition to background statin and ezetimibe therapy. Next slide. With the caveat, again, that this is a brief overview of the methods. I'm a mathematical modeling nerd. I'm happy to talk about this all day, and I'm happy to entertain additional questions. But the quick overview of the model is as follows. We're looking at adults with established ASCVD, again, the general population with some subgroups, those that are statin intolerant, those who have heterozygous FH, and folks with a recent history of acute coronary syndrome as a marker of a more severe form of disease because they're at risk in the first few years after they've had an acute coronary syndrome or an MI. We're looking at bempedoic acid and ezetimibe as a combination pill versus inclisiran, both separately compared with maximally tolerated statin plus ezetimibe. We're not comparing these two new therapies to each other. I should also clarify that the reason we decided to use the combination pill, the bempedoic acid ezetimibe, rather than looking at the individual drug, is that we were informed that the combination pill is the same price as the individual drug. So essentially, you're getting more bang for the buck with the combination pill. The outcomes we're interested in are clinical outcomes, major adverse cardiovascular events defined as an acute coronary syndrome. Again, acute coronary syndrome is a term that includes myocardial infarctions or heart attacks, as well as unstable angina. We include stroke and cardiovascular death, the kinds of events that you would expect to be reduced based on cholesterol management. We're interested in survival, both life years and quality adjusted life years. We're interested in direct medical costs or healthcare related costs, though in an alternative analysis, we also include productivity losses. And the ultimate goal is to ascertain the incremental cost effectiveness ratio as dollars per MACE or life year gained, quality adjusted life year gained, or equal value of life years gained, which is the metric that ICER has increasingly adopted. Next slide, please. We're looking at all costs and benefits over the patient's lifetime in the United States, again, primarily from a healthcare sector perspective, as well as a modified societal perspective that includes some productivity losses. We're modeling in annual cycles. Patients can have more than one event in each year, but that's the unit of time. and future costs and outcomes are discounted at 3% per year, as is recommended by current guidelines. Next slide. The events we're looking at are elective coronary revascularization, so percutaneous revascularization with stents or coronary artery bypass grafting, acute coronary syndromes, whether they're medically managed or they undergo urgent revascularization with stenting or bypass surgery, stroke, both ischemic and hemorrhagic, deaths from cardiovascular causes, and deaths from non-cardiovascular causes. We allow individuals to have multiple events in a given year, and of course over a lifetime, but you can only die once. That was a joke. Next slide. Baseline characteristics, individuals start at 66 years. The LDL, now these are individuals who are assumed to be on maximally tolerated statins and ezetimibe, so it's at 88 milligrams on average per deciliter. We assumed statin intolerance of 10%. We varied this in sensitivity analyses. If you're assumed to be statin intolerant, then the control arm only receives ezetimibe, and the intervention arm receives ezetimibe plus the intervention drug as appropriate. There are a few exceptions to these above assumptions. Again, the statin intolerant individuals, because they're not on statins, they're only on ezetimibe, they start out with a higher LDL, and per the conversation we just had recently, that means they have higher baseline event rates because they're starting out at higher LDL. Heterozygous FH individuals, we made two assumptions. One is, of course, they start out with a higher LDL, 139 milligrams. This is assuming they're already on a statin and ezetimibe. And because they have a lifetime exposure to high cholesterol, these individuals are at risk for earlier coronary events. And so even in the secondary prevention population, we assumed a younger population starting at 62 years. Next slide, please. Again, key assumptions here, based on one's clinical history, which is I've had an MI in the past or a stroke in the past or both, that helps determine quality of life, costs, and future risk of events. Again, if there's a temporal component also, that if you've had an MI in a given year, you're at increased risk for a recurrent event in the following year, etc. Patients, we talked about this, if you're statin intolerant, you have a higher baseline LDL, so you have higher absolute risk of events. And similarly, folks with heterozygous FH, we assumed that they had a 50% higher event rate than the general population with established ASCVD, and varied that in sensitivity analyses. Next slide, please. It's both a blessing and a curse to have to model without hard outcomes data. So we have to make a series of assumptions about what's happening. So first assumption is that the real world population will replicate the LDL reductions that were seen in the trial. Next is if you reduce the cholesterol, it will actually reduce major adverse cardiovascular events. We assume that on a per millimole per liter reduction in LDL, these drugs will behave similar to statins in the base case, and in the sensitivity analysis for inclisiran, we made that assumption of what if it's actually not quite like statins, but like what was observed in the PCSK9 inhibitor trials. Again, based on what we just heard, you either believe that the data from the PCSK9 trials is an artifact because they only had 2 years of follow-up, or there's something different between statins or PCSK9 inhibitors. In either case, we assumed in the base case that the reduction would behave like it did for statins, and then we tested that in sensitivity analysis. And that becomes important when we see the results. And then of course, just like effectiveness, safety would be reproduced in the real world. So for bempedoic acid, we assumed some increase in gout. For inclisiran, we assumed injection site reactions. We did not for bempedoic acid include tendon rupture. Next slide, please. The primary mover in all of this, as you can imagine, is what is the effect on LDL cholesterol. And these are our assumptions. They came from the clinical review you just heard from Dr. Lin. we assumed that the effective bempedoic acid is variable depending on whether you are on background statin therapy or not. So if the patient is on background statin therapy, it's an approximately 18% reduction in LDL. If they're not on background statin therapy, they get a larger reduction in LDL, almost at 25%. Inclisiran, again, based on the randomized trials, we saw no interaction based on background statin therapy, so we assumed that it would have the patient's LDL cholesterol. Next slide. Each of the states is associated with its own quality of life. As you are aware, there are various ways to ascertain one's quality of life, and this is one of the sources that we used from the Global Burden of Disease. There are other sources that we tested, and we reported sensitivity analyses in the final report, in the report that you've seen. Next slide, please. About drug costs. So both of these are relatively new. Bempedoic acid has been on the market, or is on the market already, but doesn't have good net pricing data available because it's so new. So we used the federal supply schedule price, which is the price to the federal purchasers, as a stand-in price for both these drugs. For bempedoic acid ezetimibe, that was a net price of $2,800 per 56 per patient per year. For inclisiran, it's about $5,600. Now, inclisiran is not even on the market, so this is very much a placeholder price, assuming that it's the same price that the existing PCSK9 inhibitors are available at. I should also add in that for inclisiran, there's a third loading dose that happens in the first year. The model actually incorporates that price in the first year of the runs. Next slide, please. Adverse events, as I mentioned, for bempedoic acid, we modeled gout. So individuals, both of these are fairly rare, but individuals who have gout get the disutility and the cost. Similarly, individuals who have injection site reactions with inclisiran, they receive a small disutility, but these are minor injection site reactions, and we assume they're not associated with any cost. Next slide. So here's the fun part, the results. In the first 4 years of the model, individuals had major adverse cardiovascular events at the rate of 5 per 100 person years. So this is a fairly high risk group of people. I mean, if you think about primary prevention cutoffs, we're thinking 7.5% at 10 years, et cetera. These are individuals who are having 5 major adverse events at 100 years. This is a secondary prevention population that is at pretty high risk. This includes combinations of fatal and non-fatal acute coronary syndromes, strokes, and deaths from cardiovascular causes. Next slide, please. So I'll walk you through this. We'll first talk about bempedoic acid ezetimibe. We'll go through all those results, and then we'll turn our attention to inclisiran. So we can focus first on the bempedoic acid issue. On average, these are individuals who are living about 15 years from when they get into the model. And the incremental survival is about 0.18 years, or about 0.17 QALYs. That's about 3 months of additional life for a treatment with this new drug. This results in incremental costs. So there are costs associated with the new lipid-lowering therapies. There are some savings from cardiovascular events that are averted from fewer MIs and strokes. But as you can see, the net change is still an increase in cost of about $31,000. And the ICER per QALY gained is about $186,000. When we use the equal value of life year metric, it's $168,000, but still above a presumed threshold of $100,000 or $150,000 per QALY. Next slide, please. And then in probabilistic analyses, where we vary all of the input parameters simultaneously in a probabilistic manner, we found, next please, that in that intermediate zone of $100,000 or $150,000 per QALY, there were only fewer than 10% of simulations that would be cost effective. So our findings are pretty robust to uncertainty in the input parameters. Next slide. Now, if you have, there's the one way to improve an ICER, so to speak, or to improve the economic value of a drug is to look at higher risk populations. And so here we are with the base case on the left, and we go into higher risk populations. Heterozygous FH, again, this is not the universe of heterozygous FH, this is secondary prevention heterozygous FH. You can see that the ICER improves substantially because of the high risk and the higher starting LDL in this population. Similarly, in the statin intolerant population, and this is an interesting fact, as you know, you saw this coming based on what you've heard so far, is that because statin intolerant individuals start out with a higher LDL and they receive a larger risk reduction, even a relative risk reduction in LDL from the bempedoic acid, that combination means that the ICER substantially improves in the statin intolerant population. And surely it does so in the recent ACS population as well. Next slide, please. Next slide. Yeah, so you are familiar with how tornado diagrams are performed. Now, unlike the probabilistic analysis where everything is varied simultaneously, here we're varying one variable at a time and leaving everything else at the base case. And to me, this is a useful exercise to identify the top 3 or 4 things that matter in the model. And you can see consistently, and I'll show you this for inclisiran, it's about risk, it's about LDL that you started out with. So like, you know, what's the risk involved? How much does that translate into benefit if you use, whether you use statins or other methods of translating LDL reduction to outcomes? But really the number one thing on the top of this is the annual cost of the drug. It is quite sensitive to the cost of the drug. If you assume a higher price, it moves, the ICER moves substantially. If you assume a lower price, accordingly, you can quickly get it under your willingness to pay thresholds. Next slide, please. So now switching to inclisiran. Inclisiran, again, as you see, the control arm survival is about 15 years. It's the same control population, so about 5 MACE rates per 100 person years. But because the LDL reduction is pretty substantial, you're having the LDL, you achieve a slightly larger benefit, an incremental survival of 0.5, which again, for coronary disease is a pretty substantial number. 0.5 translates to about 5.5 months of increased quality adjusted survival over the course of the lifetime. There is an improvement in quality of life. Again, substantial increase in costs, which is almost entirely due to the drug costs. You have some savings from cardiovascular costs, but you have quite an additional increase in drug costs of $68,000 over the lifetime, which produces an ICER of $157,000 per QALY or $142,000 per expected value of life year gained. Next slide, please. Again, probabilistically, this time you see that 0% of the simulations are cost effective next at 100,000 per QALY, but a third of the simulations are cost effective at $150,000 per QALY, again, suggesting that our results are pretty robust to uncertainty in the input parameters. I should clarify again, this is assuming the placeholder price of $5,600 per year, which, as you've heard before, is based on the PCSK9 inhibitors. Inclisiran is not currently available in the market. Next slide, please. Similar approach, subgroup analysis of higher risk patients. You have the base case on the left. If you look at heterozygous FH, you see a dramatic improvement in the ICER because of the fact that they start out with higher LDL and they have higher event rates from that lifetime accumulation of LDL. Statin intolerant patients also have an improvement, less so than in bempedoic acid, but you see a big improvement there, as do individuals with recent ACS. Next slide. And this is that sensitivity analysis that goes back to the question that came up earlier. What is actually our best guess of the estimate of effectiveness for inclisiran? If you assume that the LDL, that halving of LDL you see with inclisiran will have the same effect as statins, that's all of our base case analyses. If you say, no, actually, I think that with the effect with inclisiran will be similar to what was observed in the clinical trials of evolocumab and alirocumab, which are the currently approved PCSK9 inhibitors, then you can see that the ICER jumps because the effectiveness assumption based on those 2 short-term trials becomes, is that the drug is less effective. And so you see that the ICER then jumps to over $500,000 per, or about $500,000 per QALY. Next slide, please. The limitations, I hope I've been transparent about the limitations of the model the whole way through. We're basing this on LDL reduction alone. There's not outcomes data. There are many statin intolerant patients who are able to tolerate a low dose of statins, which will modify both the LDL and the effectiveness of bempedoic acid. There are other side effects of the drug that may appear with longer follow-up that we haven't captured. We're driving off of the trials that are available to us. And we did not examine primary prevention models, populations. So these results should not be extrapolated to them because they typically have lower event rates and therefore lower absolute benefits. The one exception to this is heterozygous FH, where even in the primary prevention population, they have higher rates, but we did not incorporate them into the model. Next slide, please. We received a ton of public comments and we're deeply appreciative because it definitely made both the model and the discussion better than where we started. We've already addressed the issue of including ezetimibe in the comparator despite low uptake in the general population. For us, the question was about value rather than trying to force people through step therapy. We talked about the uncertainty in quality of life inputs. It does matter. If you start out with a lower quality of life, you might end up with less value from a drug that prolongs your life. The effect of dosing on long-term adherence is an unknown, as you can imagine. That's an issue, and out-of-pocket costs again may vary considerably and may affect long-term adherence. Next slide, please. So in conclusion, assuming that these new lipid-lowering therapies have the same effect on outcomes as statins, bempedoic acid and ezetimibe would not meet conventional cost-effectiveness thresholds at current prices, except in individuals with statin intolerance or in some form of higher risk. Inclisiran would meet cost-effectiveness thresholds at the placeholder price, but only if its cost-effectiveness is similar to statins, not if it's similar to that observed in the PCSK9 trials. Risk matters. That's true for all modeling exercises. And we look forward to additional data based on forthcoming trials. Next slide, please. We'll stop there, and I don't know if we have time for questions.
Well, thank you, Dr. Kazi. Again, excellent presentation highlighting the main issues with the modeling and results. I'm actually going to ask if there are any very pressing questions, and we will try to adjust our break to ensure that we have questions addressed, maybe after public comment if there is no pressing questions. Okay, I'm not seeing any. Maggie, if you would like to put the slides up. Now we would like to welcome comments from manufacturers and the public comments. And we're going to start with, I believe, Joaquim Cristino. Please introduce yourself and let us know if